As shown in the introduction, the phrase, mitochondria dilemma, describes the conflict of mitochondria requirement in red blood cell differentiation. The question is, how do red blood cells overcome this dilemma? In other words, when do red blood cells determine mitochondria removal? Dr. Kazuhiko Igarashi's group at Tohoku University previously found that when mice were fed a low-iron diet, mitochondria removal occurred insufficiently. This finding raised the possibility that mitophagy depended on an appropriate amount of iron or heme. In this paper, they investigated the relationship between iron, heme and mitophagy more precisely. The authors utilized a murine erythroleukemia cell line. MEL cells proceed with the RBC maturation process when treated with dimethyl sulfoxide DMSO. So, DMSO-treated MEL cells accumulate heme. 6-phenylacetone SA is an inhibitor of heme synthesis. In spite of DMSO treatment, cells fail to accumulate heme when SA is present. The authors examined what occurred in the maturation process when heme accumulated insufficiently. DMSO treatment leads to the upregulation of GATA1, the critical transcription factor governing red blood cell maturation. Treatment with SA diminished the GATA1 upregulation induced by DMSO. Similar results were obtained for another transcription factor TAL1, the globin genes, heme metabolic genes and mitophagy-related genes. These data show that inhibition of heme biosynthesis compromises the RBC maturation program. The authors examined the relationship between heme and mitochondria abundance by using flow cytometry. Upon DMSO treatment, percentage of mitochondria-negative cells were increased, indicating that DMSO induced mitophagy. However, cells treated with DMSO and SA retained mitochondria. This result shows mitophagy in MEL cells requires heme synthesis. Conversely, the authors added heme to MEL cells and measured mitochondria abundance. Here, DMSO treatment was not done. Heme treatment resulted in an increase of mitochondria-negative cells. Altogether, these data indicate that heme has a regulatory function for mitophagy. Finally, the authors hypothesized that heme regulates mitophagy by affecting GATA1 function. Then, they tried to identify possible genes controlled by GATA1 for mitophagy upon DMSO treatment. They used public data of GATA1 ChIP-seq and RNA-seq from MEL cells, and merged the results with the known autophagy-related genes. As a result, they picked up 25 autophagy-related genes directly targeted by GATA1. Importantly, these genes included ATG4D, a vital regulator of autophagy, and STK11 that promotes autophagy in response to environmental conditions. The authors confirmed by RT-qPCR that ATG4D and STK11 were upregulated in the red blood cell maturation, but heme synthesis inhibition by SA suppressed the upregulation. These results suggest that heme enhances the expression of GATA1 targeted autophagy genes such as ATG4D and STK11. Now the conclusion of the paper. Heme is essential for the induction of the red blood cell maturation program. Heme enhances the expression of GATA1 regulated autophagy genes such as ATG4D and STK11 and induces mitophagy. Mitochondria generate heme, and then, when sufficient heme is accumulated, heme promotes mitophagy. This research must be a clue to solving the mitochondria dilemma. However, it is still unclear how heme affects GATA1 activity, and the authors are now eager to uncover the mechanism. For the details, please find their paper in PubMed. Thank you for your attention.